but are we really getting towards the size of repair that we need in humans? So this is a great slide that came from a National Geographic magazine. And it's meant to give you an idea that, well, if we repair the mouse brain, are we going to in any way get close to repairing the human brain? Just imagine the size of the mouse brain on top of the human brain. However, I have to tell you there's encouraging data that says you can actually repair the entire mouse brain with human cells. And this was work done, I'll show you some slides. So this is where we are and where we're going. Right, that's the goal. So there's some brilliant work done by a friend of mine, Stephen Goldman, was at Cornell, now at University of Rochester. And what they did was, and again, just to reduce this to its relatively simplest level, they took human cells that were derived from spontaneous aborted fetuses. So these are human cells. And they transplanted them into a mouse that has no myelin. The mouse is called the shiver mouse. It shivers because of the lack of myelin. And then they they followed the mice for periods of time thereafter. Now this mouse would die regularly at about 140 days because of its genetic deficiency in myelination. All of the work that we and others had done in transplanting cells had produced microscopically large areas of myelin, but they hadn't changed the basic clinical phenotype of that animal. We hadn't extended their lifespan in any way. We hadn't improved their behavioral abnormalities. In the Goldman studies, what they showed was these are graphs of the survival, and in brief, the red lines are the animals that got the transplants. On the bottom line, you see the time of death. You see untreated animals, the control animals. The animals that received the cells, the human cells, actually lived for months, if not up to a year of age. So the very important first part of the study was it showed that the cell transplantation in a mouse brain, in fact, could cure the mouse, as it were, cure the genetic defect and allow the mouse to live. Now, what they did, and I put this slide up to sort of amuse you a little bit, but give you some background. Many people have been using for a long time this technique to follow cells. And what it is these mice are not extraterrestrials, though you might think they are. They are getting a fluorescent light shone on them. And two of them are bright green. And the reason that they're bright green is that they were genetically manufactured to express the jellyfish protein, green fluorescent protein. Ever in a boat in the sea, and you look when the moon is shining, you'll see the jellyfish are fluorescing. It's a beautiful sight, especially if you're there with your girlfriend, wife, whatever. Looking back, I remember in Scotland yachting and this happening. It was just a wonderful happening many years ago, and you could see the jellyfish. Well, I won't go into any more beyond that. Anyway, the point is that the jellyfish gene was isolated, the GFP gene, and these mice were generated to every cell in the mouse expresses the green fluorescent protein, hence the GFP nature. It doesn't do anything to harm the mice, but they're just little green mice if you shine the fluorescent light on them. So what we do, and others do, and Goldman did, is that we don't take this approach, but they take their human cells and they induce them to express the green fluorescent protein, and here it is in the brain. So you see that the brain transplants in different mice 20 through 52 weeks strongly expresses this GFP signal, and that indicates that the cells have migrated throughout the entire brain. And then if you take it a step further, you see the labels. It's a little different here, but what we have on the right-hand side is a stain for human nuclei. So in fact, what's happened, this is a mouse brain that's basically been taken over by human cells. And these human cells are making now not the GFP protein that is indicated with the green signal, but the missing protein in this mouse's brain called myelin basic protein. So what they did was basically make a normal brain with human cells, a human mouse brain. So it indicated to you that if we can do the same thing in MS, and actually the challenge in MS is perhaps indicated to you, we're talking about repairing single areas of damage, not the entire brain. It's highly feasible given this kind of technology. So where are the cells going to come from? I couldn't get away without telling you something about embryonic stem cells. first written up by Thompson now 11, 12 years ago. So you know about this. The embryonic stem cell, of course, comes from the inner cell mass of the embryo prior to implantation in the womb. These, of course, again for background information, these are from embryos where the parents have decided that they're no longer going to keep them for further the generation of further family members or children. They're going to be destroyed, and they're then used for research purposes, and embryonic stem cells are derived from those. And one of the big challenges that we've been working on for 5 years is taking those cells as the building blocks for making oligodendrocytes in huge numbers, because that's the whole point about ES cells. They will divide indefinitely in culture and be able to give you the numbers you need for brain repair. And I have to say, in summary, we're not quite there yet. And one of the big problems, of course, again, is, as you all will know, I think, is that there are ethical questions about doing this. I personally have no ethical concerns, quite the opposite. But there are ethical concerns, and it does make it difficult. So I think one of the big frontiers in all of this work is, are there other ways of deriving cells that are like embryonic stem cells, that don't involve all of the ethical backgrounds and concerns that people have? And indeed, I'll tell you about this. So the idea is that you don't have to use embryos as the source for these cells. You can actually take mature cells, for example, skin cells, skin fibroblasts, isolate them in a skin biopsy, and then you do the following. You take these fibroblasts, and it was done first with mice and then with humans, and you insert genes into the adult cells that are associated with the embryonic state. You try to make the cells think that they're now embryonic cells. And lo and behold, if you get the right mix of genes inserted into these cells, they do indeed become what is more or less a bona fide embryonic stem cell. Incredible science. It was done a number of years ago by this fellow here in the middle of the group, by Shinya Yamanaka, surrounded by, I think it's about 30 in the main young scientists between the ages of, I would say, 25 and 30, who probably worked 16 hours a day, 7 days a week to do what he did. And you know, these genes, I said 4, there could be up to 30, and the combination, if you imagine then mathematically, the combination of these genes is so huge. So it's a shotgun approach that they used to isolate the combination of genes, and it was a brilliant piece of science. At first people didn't believe him, but it actually, I skipped a slide, but it unleashed a huge scientific race that is still going on to be, as it were, the first there. To be able to take a single gene, to be able to take a gene that didn't induce the cell to become a tumor. That was one of the problems with the original Yamanaka genes. This is now called induced, these cells are called induced pluripotential cells. Pluripotential means they can make any cell of the body. And it's pretty well where most of the action is in all of this research. So from an MS perspective, the idea is you could take a skin biopsy, grow fibroblasts, differentiate those cells towards an embryonic stem cell. In other words, put them back in time to make them think they're immature. And then from those cells grow oligodendrocytes that are genetically yours. So they cannot be rejected. This is your own cells going back into you. And this is probably where the future of regenerative medicine lies. It's all about technology. It's all about making this a safe procedure, using genes that are inexpensive to isolate. And it's very possibly in 10, 20 years going to be the way forward for treating many diseases. You simply get a skin biopsy, make these cells immature, differentiate them towards the cell of choice, and away you go.